Okay, let's get started on week 4. We're still talking about cells, but now we're going to focus on the nucleus of the cell and genetic material and how that leads to cell functions. So, DNA, as you probably are aware, it's deoxyribonucleic acid. And I'm sure you've heard it's a long thread like molecule with a uniform diameter but a varied length. There are 46 DNA molecules, which we call chromosomes, in most human cells. And the average human DNA molecule is about 2 inches long, but it's very, very thin and not visible unless it's coiled up. DNA and other nucleic acids are polymers of nucleotides, and each nucleotide consists of 3 major components: a sugar, a phosphate group, and a nitrogen base. In the case of DNA, that sugar is deoxyribose, and in DNA, the bases we have are A, T, C, and G. 2 of those are purines, which have a double ring structure, that's adenine and guanine, and 2 of them are pyrimidines, cysteine, cytosine, and thymine in DNA, and uracil in RNA. These only have a single ring structure. So we have our phosphate group, which can be 1 to 3 phosphates, deoxyribose in DNA, that would be ribose in RNA, and this one has an adenine group on it. You can remember the phrase pure silver tag C and what it means, you will know a lot about DNA. The purines are Ag. Ag is the symbol for silver on the periodic table. T binds with A, and G binds with C. Purines have 2 ring structures, pyrimidines have 1 ring structure. Cytosine and thymine are found in DNA. Cytosine is found in both DNA and RNA, but in RNA we use this modified uracil instead of thymine. The double helix shape of DNA resembles a spiral staircase. Each side piece is a backbone of phosphate groups alternating with deoxyribose, so phosphates bind to the sugar. The step-like connections, if you think of it like a ladder, a twisted rope ladder, are base pairs, and it's those base pairs in which A binds with T and G binds with C. The purine will pair with a pyrimidine that gives us the DNA sequence, and this would be the law of complementary base pairing. The sequence of one strand governs the sequence of the other, which is going to be the basis for how DNA is replicated. The essential function of DNA is to carry instructions, to carry information. And those pieces of instructions we're going to refer to as genes. Those genes specify the sequence of amino acids that will go into a protein. So that gene is a segment of DNA coding for a synthesis of a specific protein. It's been estimated that humans have approximately 20,000 genes. That would only account for 2% of the total DNA, leaving the other 98% non-coding DNA. However, don't think of that as just filler. It does have a role. It plays a role in chromosome structure. It helps to regulate gene activity. It's not what used to be referred to as junk DNA. Chromatin is the form of DNA we find in the cell most of the time. DNA in the form of chromatin is unwound. It's wrapped around some proteins called histones. 46 of them in the cells. They're 6 feet long, packed into a cell nucleus, 5 microns in diameter. It's so slender you can't see it with a light microscope. It does have a granular look appearance under the electron microscope. Those histones are the proteins with the DNA winds around. They cluster in groups of 8 molecules. DNA molecules wind around the cluster, sort of like thread around a spool. Creates a granule called a core particle. And chromatin consists of thousands of repeating core particles. Chromatin forms complex loops and coils. It's 150 times thicker and 1,000 times shorter than the naked DNA. Each chromosome is packed into its own region of the nucleus called a chromosome territory and permeated with channels that allow regulatory chemicals to have access to the genes on that chromatin. In non-dividing cells, the chromatin isn't static. It's not just sitting there. Its structures and locations change moment to moment according to the genetic activity of the cell. Genes will get turned on and off all the time. As the cell prepares to divide, it's going to make a copy of all the nuclear DNA. Each chromosome would then consist of 2 parallel filaments of identical DNA, which we refer to as sister chromatids. The chromatids are joined at the constricted centromere, usually around the middle of the cell, middle of the strand. And there are kinetochores there, which are proteins on each side of the centromere that play a role in cell division. So here we have an electron microscope image of a chromosome, which was chromatin when it was unwound, and an artist's rendering of it. You can see the parts. Ribonucleic acids, the other form of nucleic acid, comes in several different forms. Their general structure is they have a sugar, in this case ribose, which does have the oxygen that deoxyribose is missing. In RNA, the bases are going to be A, U, G, and C, because uracil is going to replace thymine. They have a single nucleotide chain, not a double helix, except for short regions. They do have three-dimensional structure. They're smaller than DNA and can have less than 100 or just over 10,000 bases per molecule. They function primarily in the cytoplasm, and the 3 that are most important to us at this level are messenger RNA, ribosomal RNA, and transfer RNA. So a gene itself is difficult to define. Classically, a gene was a segment of DNA that coded for a particular protein. But we have millions of proteins and only about 20,000 genes. So currently, we think of it more as a gene as an information-containing segment of DNA that codes for the production of a molecule of RNA that most often plays a role in synthesizing one or more proteins. One gene, one polypeptide is a good way to think of it. The amino acid sequence of a protein is determined by the nucleotide sequence of the DNA. 46 human chromosomes come in 2 sets of 23. We get 1 set from each parent. All the DNA in the 23 chromosome set is known as the genome. Approximately 3.1 billion nucleotides in a typical human genome. Individual variations come from single nucleotide polymorphisms. Those are nucleotides that can be different between individuals. Genomics is the study of the whole genome, how the genes and non-coding DNA interact to affect structure and function of the organism. And a future of medicine, rapidly approaching, is genomic medicine, application of the knowledge of the genome to predict, diagnose, and treat diseases. This was likely to have great implications for many disorders: cancer, Alzheimer's, schizophrenia, obesity, acquired immune deficiency syndrome, and tuberculosis. So the body can make millions of different proteins. That's called the proteome. It does so from just 20 amino acids. They are encoded by the genes and which are made of 4 nucleic acids or nucleotides, A, T, C, and G. So there is a genetic code. That genetic code is the system that enables 4 nucleotides to code for all of the different amino acids. The code is written in groups of 3. A base triplet, so the sequence of 3 DNA molecules that stands for 1 amino acid would be a base. Codon would be the 3 base sequence in the messenger RNA. There are 64 possible codons, 64 combinations of 3 if you have 4 to choose from, and they are used to represent 20 amino acids. 61 of those do code for an amino acid. The other 3 are stop codons. The start codon, AUG, which also codes for methionine, begins the amino acid sequence of the protein. The stop codons include AUG, UGA, and UAA. They are sort of like the period at an end of a sentence. So in all body cells, except sex cells and some immune cells, genes are identical. Different genes are activated in different cells. Any given cell is using about 1/3 to 2/3 of its genes at any time, and the rest remain dormant or may be functional in other types of cells. When a gene is activated, messenger RNA is made, which serves as a code for a particular protein. So DNA is converted to RNA, copied to RNA. RNA is translated into a protein. Process of going from DNA to messenger RNA is called transcription. That occurs in the nucleus. From messenger RNA to protein is called translation, and that occurs in the cytoplasm. So transcription is the copying of genetic instructions from DNA to messenger RNA. It utilizes an enzyme called RNA polymerase. That enzyme will bind to a start sequence in the DNA, and it will open up the double helix. RNA polymerase will read bases from one strand of DNA and build a complementary strand of RNA, which means that when it gets to a C on the DNA, it will add a G of RNA. When it finds a G on the DNA, it will add a C of RNA. When it finds a T on the DNA, it adds an A of RNA. And where it finds an A, it does not add a T, it adds a U of RNA. Remember, there's no T in RNA. That transcription continues, and RNA polymerase will then rewind the DNA helix behind it, allowing the gene to be transcribed by several polymerase molecules. There's a terminator sequence at the end, and the pre-messenger RNA, especially in eukaryotes, is still immature. It has to be processed before it is ready for translation. So enzymes within the nucleus will remove introns from the RNA and splice the exons together. One way to get different proteins is to use a process called alternative splicing, which you don't use the same exons every time. And that's how one gene can code for more than one protein. Once you have that mature messenger RNA, it goes to the cytoplasm, and it brings into play the other two types of RNA, transfer RNA and ribosomal RNA. So the ribosome is going to read the messenger RNA code and form a protein. Messenger RNA has the instructions. It has the sequence that tells you which amino acid goes next to which amino acid. Transfer RNA is the translator molecule. It delivers a single amino acid to the ribosome based on the anticodon of the codon of the messenger RNA. So transfer RNA reads the codon, and it has on its sequence an anticodon, 3 nucleotides that are complementary to the messenger RNA codon. That ribosomal RNA, and then ribosomal RNA makes up a big portion of the ribosome. They have a large subunit, a small subunit. They can be free in the cytosol, they can be on the rough ER, or they can be on the nuclear envelope. And there are 3 important sites on that ribosome, the E site, P site, and A site, that are involved in this process of translation. So here is a transfer RNA. Down at the bottom, we see a anticodon, UAC, so this would bind to the codon on messenger RNA, AUG, sorry. Translation occurs in 3 main steps. So the initiation begins when the ribosome assembles with messenger RNA in the cytosol. Small subunit binds to the leader sequence of the messenger RNA. Large subunit joins when the AUG codon is reached. Protein synthesis begins. a transfer RNA carrying the amino acid methionine comes in. In the next step, our transfer RNA with the next amino acid binds to the A site. So now we have a methionine and another amino acid bound to the A site. The ribosome will then create a peptide bond between those two amino acids and move down one codon. So the growing RNA is now attached to the transfer RNA that is in the P site. When you reach a stop codon, termination begins. It's a protein release factor binds to it, and ribosome disassembles and dissociates from the messenger RNA. Proteins that are destined for lysosomes or secretion are modified by the ER. The ribosome will dock with the ER and will actually synthesize that protein into the endoplasmic reticulum. Well, there it will be packaged, modified, and transported in transport vesicles. Multiple ribosomes in clusters called polyribosomes can translate the same messenger RNA molecule simultaneously. So DNA, again, is a double helix. There are 7 base triplets on the template strand of DNA in this case. The corresponding codons of messenger RNA are transcribed from the DNA template. So we have an A here, which would be a U. We have a T here, which would be made into it would add an A RNA. Here you have an A DNA. You would add a U RNA, and so on. Those groups of 3, those codons, will bind to anticodons on the transfer RNA, and on the transfer RNA are the amino acids that are going to be put together, finally giving you the amino acid peptide chain. There is post-translational modification in many cases in which proteins assembled on the endoplasmic reticulum surface goes through the pore. The enzymes in the ER modify the protein, sometimes removing amino acid segments, folding the protein, stabilizing it with disulfide bonds, and adding other things to it. Proteins would then pinch off from the endoplasmic reticulum in a transport vesicle coated with clathrin proteins. Clusters of those vesicles fuse and form a new Golgi cistern on the cis side of the Golgi apparatus. And that new cistern migrates through the Golgi to the opposite side, or trans side of the Golgi, where it breaks into vesicles containing the cell products, and those vesicles become or go to whatever they are going to be: lysosomes, secretory vesicles, or other types of vesicles. So protein processing and secretion begins in the endoplasmic reticulum. From the endoplasmic reticulum, it moves to the cis side of the Golgi complex, and then finally to the trans side of the Golgi complex, where it is delivered to where it needs to go. Lysosomes here, secretory vesicles here. Genes can be turned off and on. Cells can turn some genes permanently off. Liver cells don't make hemoglobin, for example. Cells can turn genes on only when they're needed. The level of gene expression varies from day to day, hour to hour, and that can be controlled by chemical messengers such as hormones. For example, in response to prolactin, mammary gland cells turn on the gene for casein protein only when breast milk is being produced. Cells can also synthesize things like glycogen, fats, steroids, phospholipids, pigments, and other compounds. They don't have genes for these, but their synthesis is still under the direct control or the indirect control of the genetic control, because they are all produced by enzymes, and those enzymes are coded for by the genes. So for example, if you were to produce testosterone, a cell of the testes would take in cholesterol. An enzyme would convert that cholesterol to testosterone. That will only happen if that enzyme to convert cholesterol to testosterone is present, and that will only happen if the gene for that enzyme is turned on and active. So genes can greatly affect complex structures such as behavior, aggression, and sex drive in ways that we don't completely understand. Before a cell divides, it has to duplicate its DNA so it can give a complete copy of all its genes to each daughter cell. Since DNA controls all cellular function, this replication process has to be exact, and the law of complementary base pairing enables us to predict the sequence of one DNA strand if we know the other. So, replication, not the same thing as transcription. Transcription, you're making RNA. Replication, you're making DNA. We can break replication down into 4 steps. First of all, we will unwind the helix from the histones, then unzip small portions of the helix by an enzyme called DNA helicase, separate the strands, which will form a replication fork. An enzyme called DNA polymerase will then move along each strand, read the exposed bases, and synthesize the complementary strand. One of those strands, it can go right along because it's in the correct direction. On the other strand, it has to go in little segments. And then an enzyme called DNA ligase will come in and tie those segments together. So what we have is described as a semi-conservative replication. Each new DNA molecule will have an original strand and a newly synthesized strand. New histones would then be synthesized and used to organize its DNA strands into nucleosomes. So we open up the DNA at the replication fork. DNA helicase unwinds the DNA as it goes, and then DNA polymerase comes along, and you can see this is the leading strand. It can just run the whole length. In the lagging strand, we get a little bit of gaps, and those are filled in by DNA ligase. Sometimes polymerase makes mistakes. It's a good thing it makes mistakes, otherwise we'd all be whatever that first life form was that formed on the planet 4.5 million years ago. We have DNA damage response, the mechanisms in place to correct replication errors. DNA polymerase double checks the new base pairs, tends to replace incorrect, biochemically unstable pairs with more stable correct pairs. And that, in the end, results in 1 error per 1 billion bases replicated. Those changes are known as mutations. They can be due to replication errors. They can be due to environmental factors. Some mutations cause no effect. Most mutations are detrimental mutations, and some mutations are favorable mutations. Genetic variation within one's own body due to these mutations and replications and error is called genetic mosaicism, because you have different DNA in those cells that result from that cell that made a mistake copying. Cell cycle includes interphase and the mitotic phases. Interphase can be divided into 3 subphases: gap 1, synthesis, and gap 2. In the mitotic phase includes prophase, metaphase, anaphase, telophase, and cytokinesis. Can map this out. So a cell, when it is first formed, is in G1, first gap phase. It's going about its normal business. If that cell is going to divide, it will then move into the S phase, undergo DNA replication, as we just talked about, make a complete copy of its DNA, and then go into the second gap phase, where it would continue to grow and make other preparations needed for mitosis. At that point, we're going to switch to chromosomes as we reach the mitotic phase. Those chromosomes will form in prophase, line up in the center of the nucleus in metaphase, pull apart from each other in anaphase, and split into 2 nuclei in telophase, which closely overlaps with cytokinesis, where the 2 cytoplasms separate from one another. So G1 is the interval between cell division and DNA replication. That's when a cell, as I said, is doing its normal tasks, synthesizing proteins and materials for the next phase. If the cell is going to divide, it will go to the S phase. Cells will replicate all the nuclear DNA and duplicate the centrioles. In G2, we have a gap before mitosis begins. The cell repairs DNA replication errors, grows and synthesizes enzymes you need for cell division. In the mitotic phase, you're going to replicate its nucleus, which is going to pinch, and then that cell is going to pinch into two cells and form daughter cells. We sometimes use the term G0 for a cell that will remain in G1 for permanently or for a very long time because it will never go to S. Mitosis is cell division resulting in two genetically identical daughter cells. In contrast to that, when we make sex cells, sperm and egg, we use a similar but different division called meiosis. Meiosis is a reductive division. Mitosis is an exact copy division. Mitosis is used for the development of an individual. We all started out as one single cell approximately 9 months before we were born, and that cell got to where you are today by going through mitosis trillions of times. Growth of all tissues and organs after birth, replacement of cells that die, and repair of damaged tissues. If we look more closely at mitosis phases, they are again prophase, metaphase, anaphase, and telophase. In prophase, we are preparing for dividing the nucleus. The genetic material will condense into those chromosomes. Chromosomes are easier to move from place to place than chromatin. We will end up with 46 chromosomes. There'll be 2 chromatids per chromosome. We can't take DNA out of the nucleus, so for the time being, we're going to get rid of the nuclear envelope. It's going to disintegrate. The centrioles will sprout their spindle fibers, which are really long microtubules, and those spindle fibers will push the centriole pairs apart. The spindle fibers will attach to the kinetochores of the centromeres as well. In metaphase, the chromosomes align up along a line, an equator, generally around the middle of the cell. Spindle fibers will form lemon-shaped array called the mitotic spindle. Shorter microtubules from the centrioles complete a star-like aster which anchors itself to the inside of the cell membrane, and you will have one of these on each side. So the chromosomes have lined up in the middle for metaphase. They were prepared in prophase, lined up in the middle in metaphase. In anaphase, the centromere is going to split. When the centromere splits, we now have single-stranded chromosomes. We have a chromatid. The 2 chromatids have separated from each other, but now we call them chromosomes instead of chromatids, and they will migrate towards the kinetochores. When they get far enough apart, the telophase will occur. The chromosomes will uncoil and become chromatin. Mitotic spindle will disintegrate, and a nucleus will form around each set of chromosomes. So in prophase, we prepared for the cell division by making chromosomes. In metaphase, we lined those chromatids, 2 sister chromatids joined by a centromere in the middle of the cell. In anaphase, we split the centromere and pulled the chromatids apart, now calling them chromosomes. And in telophase, we will have 2 nuclei in the cell for a brief period. That overlaps with a process called cytokinesis, in which we get a division of the cytoplasm into 2 cells. It's achieved by the proteins myosin and the cytoskeleton. Starts with a cleavage furrow, and that eventually pinches into 2 completely new cells. New cells that are supposed to be genetically identical to each other. Cell division obviously has to be tightly regulated. When we don't have regulation of cell division, we end up with what we often refer to as cancer in many cases. So cells will divide when they have enough cytoplasm for two daughter cells, after they have replicated their DNA, if they have an adequate supply of nutrients, if they are stimulated by growth factors, those are chemical signals, when neighboring cells die and open up space. Cells are supposed to stop dividing when they are in contact with neighboring cells, when nutrients or growth factors are withdrawn. They have what is known as contact inhibition. When they come in contact with other cells, they stop dividing. Cancer cells lose that contact inhibition. It's regulated by a molecular timer and checkpoints. Proteins constitute the timer. We have cyclins, which form during interphase. Their levels rise and fall through the cell cycle. And then we have cyclin-dependent kinases, which are activated by cyclins to phosphorylate other proteins. We talked about second messengers, and I told you that kinases add phosphates to things, and adding phosphate can turn on or turn off different activities. So the cyclins are there. The cyclin-dependent kinases are formed, and cyclins activate the cyclin-dependent kinases, and they will turn on and off the enzymes needed to move to the next phase of mitosis. We have them at the replication of DNA in the centrioles, in the S phase, the condensation of chromosomes, breakdown of nuclear envelope, mitotic spindle, and splitting the centromere and separating the sister chromatids. We have a start or G1 checkpoint. Those cells that don't go through that G1 checkpoint that we said we can refer to as G0 are amitotic cells, cells like nerve cells. There's a G2M checkpoint. Latent G2 determines whether the cell is able to proceed to mitosis. And there are other checkpoints, including one from metaphase to anaphase, to determine whether the cell can proceed to anaphase and separate the sister chromatids. A karyotype is a way to look at the DNA. We look at the chromosomes of dividing cells because we can see those chromosomes with a light microscope. So heredity is the transmission of genetic characteristics from parent to offspring. So your karyotype would be all 46 chromosomes laid out in order by size. There are 23 pairs of them, so you'll have 2 of each. Chromosome number 1 is chromosome number 1 because it's the longest one. A homologous chromosome is one of the 2 inherited from each parent. So you have 2 copies of chromosome number 1. A homolog you got from your mom and a homolog you got from your dad. 22 of those are known as autosomes, 1 through 22. The third pair of sex are known as sex chromosomes. In humans, a female has a homologous pair of X chromosomes, one X from mom and one X from dad. And a male has an X that it got from its dad and an X that it got from its mom and a Y that it got from its dad. Here is a normal human karyotype. You can see 2 copies of each chromosome. Diploid is the cell with 23 pairs of chromosomes. Most somatic cells are diploid. Haploid describes cells that have half as many chromosomes as somatic cells. Human haploid cells would contain 23 unpaired chromosomes, and they would be the sex cells, the germ cells, sperm and eggs. When you put a sperm and an egg together, 2 haploid cells gives you the diploid number of chromosomes. And from that fertilized egg, you will get a somatic cell. And that somatic cell can undergo mitosis to become an organism. More terminology. Genes are found on a position in the chromosome we refer to as its locus or loci. Alleles are different forms of a particular gene. Alleles are found at the same locus on homologous chromosomes. Alleles can be dominant allele, which we would represent with a capital letter. The trait would be seen. If the dominant allele is present, it would mask recessive alleles. It often produces proteins responsible for the trait. A recessive allele, represented by a lowercase, is only seen if you are homologous for that recessive allele. If you have two copies of the recessive allele. If you have a dominant allele, you'll cover up the expression of the recessive allele. Your genotype is the alleles that any individual possesses for a particular gene, and so you can be homozygous, two of the same alleles, or heterozygous, have a different allele. The phenotype is the observable trait. So an allele is expressed if it shows the phenotype of an individual, usually determined by multiple genes, but there are some genes that arise from some traits that arise from single genes. In fact, if you've, and I'm sure you have, heard of Gregor Mendel, you probably know about at least 7 single gene traits in pea plants. And we can use a Punnett square to show the possible genotypes and then predict the possible phenotype outcomes from parents of a known genotype. Parents can be healthy, heterozygous carriers of genetic diseases, and genetic counselors are there performing genetic tests and advising couples on the probability of transmitting genetic diseases. Gene pool is the genetic makeup of the whole population. In a gene pool, some genes come in 2 or more allelic forms called multiple alleles. For example, for blood types, we have the gene I, which codes for an enzyme that makes a carbohydrate, and there are 3 forms of that. IA, which makes enzyme that makes A carbohydrate, IB, that makes the enzyme that makes B carbohydrate, and lowercase I, which doesn't make an enzyme. In codominance, both alleles are equally dominant. Both are phenotypically expressed. For example, an individual with IA and IB will have blood type AB. This slide is wrong. It's not type AO, it's AB. In complete dominance, you get an intermediate phenotype, and the heterozygous shows a phenotype that's in between the extremes of each trait on its own. Hypercholesterolemia. Polygenic inheritance means that genes at 2 or more loci can contribute to a single trait. And to be honest, most human traits are polygenic inheritance. Most of the things we would be interested in, most of the phenotypes we would be interested in are due to polygenic inheritance. Pleiotropy is when one gene produces multiple phenotypic effects. And so in alkaproteinemia, you have a mutation on chromosome 3 that results in the darkening of the skin, darkening of the sclera, and urine becomes dark upon sitting. So polygenic, we have at least 3 genes that contribute to eye color. Alkaproteinuria. Some genes are carried by the X and the Y chromosome, and they would tend to be inherited more by one sex than the other. Recessive color blindness on chromosome X is an example of that. When we get to the visual system eventually, we'll see that color blindness is more common in men than women. It's a common misconception that dominant alleles must be more common in the gene pool than the recessive alleles. Some recessive alleles are the most common. Blood type O is the most common allele. Some dominant alleles are rare in populations. Polydactyly, having an extra digit on your hand or feet, is a dominant mutation, but it is relatively rare. Blood type AB is the rarest of the blood types. Gene expression is when a gene is turned on and has a noticeable effect on an individual. The amount of effect would be its penetrance, so we can have incomplete penetrance when a dominant allele is silenced by someone who carries it. That could be because of environmental factors influenced by gene expression. So, for example, genes for melanin eye pigment can only be fully expressed if there's phenylalanine in the diet. There are also epigenetic effects. Genes are expressed without altering the changes in the base sequence. They can be passed to offspring, called epigenetic inheritance. DNA methylation is an example of this. There are many epigenetic changes that are normal. Some may trigger diseases such as cancer. Epigenetics, we are still really trying to get a handle on at this point. Cancer is the uncontrolled growth of cells, which will cause tumors. And if those tumors become malignant, we refer to them as cancers. Cancers grow rapidly, tend to metastasize, which means they spread. They stimulate tumor angiogenesis, which is a growth of blood vessels in order to feed these tumors. And they are distinguished from slow-growing, encapsulated, benign tumors. I'm sure you're aware of metastases. We can treat almost all primary cancers. By the time they metastasize, it becomes much more difficult and in many cases impossible. Oncology is the medical specialty dealing with tumors. They're named for their tissues of origin, so a carcinoma would arise in an epithelial tissue, a lymphoma in lymph tissue, a melanoma in the pigment cells of the epidermis, leukemias in blood-forming tissue, and sarcomas in the bone or other connective tissue or muscle. Cancers are genetic in the sense that they are caused by mutations in the genes that control cell division. But only about 5 to 10% of those are hereditary. Most are caused by mutations that occur in somatic cells, not mutations that we pass on to future generations. During replication or exposure to carcinogens, environmental cancer-causing agents, radiation, ultraviolet chemicals, viruses, all kinds of things can mess up the way DNA gets copied when it's being copied to repair things within an organism, for example. Oncogenes cause cells to divide out of control. Tumor suppressor genes block the development of cancer, but when they are mutated, we can lose the expression of tumor suppressor genes. Cancer replaces functional tissue and vital organs. You don't really have extra space for anything in your brain, or you do, unfortunately, in your lung tissue, which is one of the reasons it takes a long time to catch lung cancer. Tumors can put pressure on other organs. They weaken immunity, and they also steal nutrients from the rest of the body. They're cancer wasting disease. Cancer cells will take nutrients away from healthy cells. Here is an example of a cancer, a Wilms tumor, shown in the white. And I believe that is going to wrap up, yes, our discussions of genetics.